Primer walking became extremely useful for sequencing plasmids, clone genes, and closing gaps left behind in larger genome projects. However, imagine trying to walk through an entire continent using this method. Sequencing a whole genome base by base in this way would be extraordinarily slow and expensive. Scientists needed another solution. Shotgun sequencing is what was done using for solving the puzzle. Now let us change the scene. Now, suppose someone gives you a 1,000-page novel and asks you to read it quickly. Instead of turning each page one after another, you tear photocopies of the book into hundreds of pieces. Many people read different fragments simultaneously, later using overlapping words and sentences. The pages are reconstructed until the complete story emerges. This is the philosophy behind shotgun sequencing. In this method, the entire DNA molecule is broken randomly into numerous smaller fragments. These fragments are then sequenced simultaneously. At first glance, the result appears chaotic. Thousands or even millions of short sequences are generated without any obvious order, but hidden within that apparent disorder are clues. Many fragments overlap with one another, and sophisticated computer algorithms identify these overlaps and align them, gradually reconstructing the original DNA sequence. The name shotgun reflects this randomness, just as pellets form a shotgun scatter in many directions. DNA fragments are generated randomly across the genome. Unlike primer walking, which relies on careful sequential progression, shotgun sequencing embraces randomness and harness computational power to create order from complexity. So, this strategy transformed biological research, and a large genome that would have taken years to sequence by traditional approaches could now be decoded in a fraction of the time. The method played a pivotal role in ambitious projects such as sequencing of the human genome, and it has become the foundation of modern next generation sequencing technologies, even. So, comparing these two strategies, we see that if primary walking is the journey of meticulous traveler, shotgun sequencing is the work of a master puzzle solver. Primer walking values patience. It moves carefully, ensuring that every step is verified before taking the next one, whereas shotgun sequencing values speed. It divides the challenges into countless smaller tasks and uses computational intelligence to rebuild the whole picture. Neither method is inherently superior. Their usefulness depends on the question being asked. When a researcher needs to verify a cloned gene or sequence, a relatively small DNA fragment with great accuracy, primer walking remains valuable. When the goal is to explore entire genome, microbial communities, or massive data sets, shotgun sequencing becomes indispensable. So, in conclusion, we can say that the story of DNA sequencing teaches us an important lesson, and science advances not only through powerful technologies but also through the creative ways of thinking. So one method taught us the importance of moving forward carefully, one informed step at a time, and the other taught us that even the apparent chaos can reveal extraordinary order when guided by logic and innovation. So, whether by walking patiently through the sequence or by assembling the scattered fragment into coherent whole, these strategies have in fact enabled humanity to read the language of life itself. And because of them, we can now understand genes, diagnose diseases, trace evolution, improve crops, and even envision a future. Where biology and technology work hand in hand for the betterment of the society. Thank you.